Go ahead.
All right.
Okay. Is my screen share okay?
Yep, looks good.
Yeah. Yeah, so let's get into session 4. So today we're going to quickly review the central dogma and genetic code, and then we're going to go into translation. We're going to look at some of the differences between prokaryotic and eukaryotic translation, and then go through the major steps of translation, which are initiation, elongation, and termination. Then we're going to do a quick summary of protein structure. There'll be some more of that next week as well. Then we're going to take a break for questions, and then we'll go into protein-related techniques. So the ones I'll be focusing on are protein purification, protein separation by SDS page gel, and immunodetection techniques. Okay, so let's get into translation. We've gone over this a few times, but the main concept of flow of genetic information is that you go from DNA to RNA to messenger RNA to protein. So we've covered transcription and reverse transcription, which is the relationship between DNA and RNA. And today we're going to focus on translation, which is how messenger RNA is translated into amino acid chains, which comprise proteins. This is genetic code, which we've already seen a few times in this class, but just want to point it out again. So the mRNA transcript is read in the form of codons. So codons are sets of 3 nucleotides. So there are 64 possible codons. These include 3 stop codons, which code for the end of termination of translation, 1 start codon, which is AUG, and that signals the start of translation. And there are 2 that are unique for amino acids, but 59 of the 64 codons are redundant. So that's one of the key features of the genetic code, in addition to being universal and punctuated. So I'll explain what punctuated means in this context in a bit. So the mRNA transcript can be read through 3 potential reading frames, depending on where you start. So there, as you can see here, depending on which nucleotide you start reading a codon at, there are 3 possible ways of translating this mRNA sequence into an amino acid sequence. So you'll notice the mRNA sequence is identical across all 3 of these, but depending on where the first codon starts, they're coding for completely different amino acids. So the start codon is sort of the one of the punctuating factors that indicates where translation should start. So just going to go over a quick overview of translation. So in translation, you start off with an mRNA transcript, and this is exported from, in the case of eukaryotes, it's exported from the nucleus. In the case of prokaryotes, it's already in cytoplasm. And then the ribosome, which is a complex of multiple proteins and ribosomal RNA, then forms a complex around mRNA, which also involves the use of transfer RNA or tRNA. And this process will translate the mRNA transcript into a polypeptide, which is a sequence of amino acids. There are 4 major steps involved in this process. So the first is amino acid activation, then you have chain initiation, elongation, which is repeated multiple times, and then you have chain termination to yield a completed polypeptide. So a key molecule involved in translation is transfer RNA or tRNA. This is an adapter molecule that reads the codons and transfers the amino acids. So it's like other RNAs, it's transcribed in the nucleus, and then it is then it undergoes processing. Specifically, it undergoes some trimming, and there's an intron that needs to be removed in most cases. And then this nucleotide stretch, which is about 80 nucleotides, it forms a clover shape. So it has 4 segments that form double helices. The key features that we want to focus on is this region right here, which is called the anticodon loop. So that is complementary to the codon that it will recognize in the mRNA transcripts. The other important feature you want to focus on is the CCAN, which refers to these last 3 nucleotides. This is where the amino acid will be bound to.
Can I ask a question? Is there just a single tRNA, or are there different tRNAs for each codon?
There are different tRNAs for each amino acid, and the major difference between them all is what the sequence of the anticodon loop is and which amino acid is going to be attached. So before a tRNA is able to start translation, it needs to be charged, which means the correct amino acid needs to be attached to it. So this is done by an enzyme called aminoacyl tRNA synthetase. We cannot pronounce things. So it reads the anticodon and as well as the sequence at the amino acid acceptor stem region, and it attaches the proper amino acid to the tRNA molecule. So for the first 2 nucleotides in the codon, the stringent accuracy is necessary for base pairs. But in the 3rd position, this is less stringent, and this position is also called the wobble base. The reason why the 3rd position, accuracy at the 3rd position isn't as important is because of the redundancy of the genetic code. So you'll notice that in many cases, there are multiple possible codons that code for a specific amino acid, and usually it's the 3rd nucleotide that is different in most of these cases. So that's why there's a potential mismatch at the 3rd position wouldn't change the amino acid sequence. Okay, so as I mentioned before, translation occurs at the ribosomes. So again, ribosomes are a complex made of proteins and ribosomal RNA. It consists of 2 subunits, a large subunit and a small subunit, which are represented here. So normally these 2 subunits are separate, but during translation, they form a complex around mRNA, which is threaded through the 2 subunits. There are also 3 sites that you should be aware of. Those are called the E site, the P site, and the A site. So that stands for, the E site stands for exit, the P site is the peptidyl tRNA site, and the A site is the aminoacyl tRNA site. So there's some major differences between prokaryotic and eukaryotic translation, and a lot of it has to do with how it's initiated. So the first major difference is mRNA structures. So prokaryotic mRNA doesn't undergo the same level of processing as eukaryotic mRNA. So just a refresher, eukaryotic mRNA has this 5' methyl 7-methyl GTP cap, and this aids in the binding between the mRNA molecule and the small ribosomal subunit. It also has a polyadenylated tail. In addition, sizes of ribosomes and RNA are different between prokaryotes and eukaryotes. Recognition of the initiation consensus sequence is also different between prokaryotes and eukaryotes. So in prokaryotes, it's the initiation consensus sequence is called the Shine-Dalgarno sequence. In eukaryotes, it's called the Kozak sequence, and I'll go into more detail about that in a little bit. There are also some differences in the initiator tRNA. So translation initiation in prokaryotes depends on this sequence called the Shine-Dalgarno sequence. Is a purine-rich ribosome binding site. So this is upstream of the start codon. So the start codon is the sequence AUG. The Schindel-Garno sequence is complementary to the rRNA that is part of the small ribosomal subunit, and this promotes binding of the mRNA to the ribosome. A single mRNA molecule in a prokaryote can have multiple start codons and multiple ribosome binding sites, or SD sequences. This is something that is not the case in eukaryotes. In eukaryotes, the recognition sequence for the ribosome is called the Kozak sequence. This sequence includes the ATG or AUG, which is the start codon. In eukaryotes, the start codon is within the recognition sequence for initiation, whereas in prokaryotes, the Schindel-Garno sequence it's usually upstream of the start codon, and it can be at multiple points in the mRNA sequence. So the start codon then is AUG, and if you look at the code here, AUG codes for methionine. The initiator tRNA is distinct from the regular tRNA coupled with methionine, however. By that I mean the actual sequence of the tRNA coupled with regular methionine is distinct from an initiator or tRNA. This initial methionine is usually removed later by a protease. And in prokaryotes, there's one difference in the initiator tRNA, that is instead of being coupled to a methionine, the initiator tRNA in prokaryotes is coupled to a derivative of methionine called formal methionine, or MET. So after initiation comes elongation. When new amino acids are added to a pre-existing polypeptide, the tRNA carrying the next amino acid binds to the A site, or amino acyl tRNA site, which is represented here. The tRNA then forms base pairs with the mRNA from its anticodon region. The carboxyl end of the polypeptide is then released from the tRNA at the P site, or peptidyl tRNA site, and it's joined to the free amino group of the amino acid linked to the tRNA at the A site. These two amino acids are now joined by a peptide bond. And this peptide bond is catalyzed by the large ribosomal subunit. This causes a conformational change that moves the mRNA along the active site, and it moves by a further 3 base pairs. As this is going on, there are also elongation factors that enter and exit the ribosome to aid in the hydrolysis reactions and to promote the conformational changes that allow the mRNA to proceed through the ribosome.
When do the tRNA bind with their complementary amino acids? Do they find them out in the cytoplasm and then go to a ribosome, or does it happen inside the ribosome?
That happens prior to getting to the ribosome, and that's carried out by an enzyme called amino acyl tRNA synthetase.
Synthetase. You have a slide on it before. You want to back up and show James again? There. This is where it happens.
Mm-hmm. Yeah, this is prior to tRNA leaving the nucleus.
That's called charging, right? When the amino acid is added to the tRNA.
Yeah.
So you need a charged tRNA to interact with the ribosome to have anything happen.
Okay, so after the mRNA sequence has been translated. Translation termination is signaled by a stop codon. So there are 3 potential stop codons here. They're coded by UAG, UAA, or UGA. So these codons are not recognized by any tRNA. Instead, what happens is that release factor protein binds to the ribosome when it binds to ribosomes that have a stop codon at the A site. And this binding causes the ribosome to catalyze the addition of a water molecule instead of an amino acid to the tRNA that's located in the P site, just the middle one. This frees the carboxyl end of the polypeptide, and the polypeptide is then released into the cytoplasm. The 2 ribosomal subunits are now separate, and the mRNA is released as well. The ribosomal subunits can now go and bind another mRNA molecule and initiate translation there. Yeah, so that's the process of translation. So are there any questions about that?
Yeah. Oh, go ahead,
sorry.
James. No, you first.
You go. I've asked a few.
Oh, go, James.
What is is there a function of the ribosomal RNA that is more than just structural? What is it doing in this whole process?
That's kind of yeah, they're involved in catalyzing the reactions that are going on here as well. It's all very complex, so I would have to look into it further. But
yeah,
it's more than just structural function.
Yeah, I think I've just doing this from memory a little bit from reading the textbook. I think that the ribosome, that most of the catalysis is done by the RNA rather than the proteins. They've done experiments, and the proteins are not near the catalytic sites, I think. I hope I'm remembering that right.
Yeah, it sounds.
I have a question, Shirley, about the FMET for prokaryotes. So the FMET instead of MET is what's added, and then some enzyme comes along and takes it off, right?
Yeah.
Do we have an idea of why that? Why why is that? Or it's not understood, or it's speculative?
Yeah, I'm not really sure if there's much known. One thing that's noteworthy is that the FMET is also used in mitochondrial and chloroplast translation of proteins from mitochondrial and chloroplast DNA genomes. So I'm not totally sure, but I find that kind of interesting.
Yeah, it's at some point there must have been an evolutionary change when eukaryotes evolved, and so there must have been a reason. Oh, look at that. Beth is looking it up in Wikipedia for us.
Another question. A lot of the schematics show one ribosome to one mRNA. Is that always the case? Is it always one at a time per ribosome, or do ribosomes translate multiple mRNAs at different parts of the ribosome?
It's one ribosomal complex per mRNA, and then once it's done, it can disassemble and then reassemble somewhere else. Yeah.
Okay.
Yeah, but the same mRNA can have multiple ribosomes on it in prokaryotes, right? Which I'm sure you're going to talk about in a minute, which is kind of rad. It's crazy. These systems are so wild. Yeah.
Not just in prokaryotes, right? Also in eukaryotes. It's just that with prokaryotes, you can get transcription and translation at the same time because there's no nucleus.
Yeah, you have that, but you can also have because of the way that prokaryotic genomes are arranged, the mRNA can have multiple start codons. So it can you can translate multiple proteins at the same time, because the ribosome, in this example, the ribosome can bind at these 3 places at the same 3 different ribosomal complexes can bind at the same time onto this particular mRNA molecule, because you see how there's 3 sites where it can bind.
I'm a little confused why the Shine-Dalgarno sequence or the Kozak sequence, why those aren't related to the start codons. Why don't they occur at the same place? It seems like maybe I'm visualizing how it happens wrong.
Well, in the consensus sequence for both of them, they are involved in binding between the mRNA and the ribosome, whereas the start codon is it's where the tRNA kind of gets involved and initiates the process of translation. So this is more about just getting the mRNA molecule into the ribosome, and this is like the actual start of translation.
So it uses the Shine-Dalgarno to grab onto it, and then once it's grabbed on, then it at the start codon it starts moving through and starts translating. Okay.
There's a scanning process, I think, where the ribosome moves forward and scans for the start codon.
Yeah. Any questions about translation?
Sorry, can I just ask one more about the Shine-Dalgarno? So how you have it represented there, it looks like the sequence isn't adjacent. It's like the sequence occurs at different parts of the strand. Why is that? Is it so it's like coiled up and all those parts attach and then, or what?
Yeah, I'm not sure. I think that this is there's just some variation across species. And like Jeremy said, there's a scanning function as well. So if this is where the ribosome binds, then it scans until it gets to the start codon. So it doesn't necessarily have to be, attached to the start codon.
So are these each different Shine-Dalgarno sequences? GGAG, GGAG.
Yeah, so these are different genes.
Okay. I see. I thought this was one sequence. Okay, I understand.
Yeah. So there are different strengths. Those sequences, right? The RBS, they're also called ribosome binding sites, have different strengths. So that will affect, like in prokaryotes, how efficiently the translation takes place.
Okay, can we get into protein structure now? Or? Okay. Okay, so there are 4 levels of either 3 or 4 levels of protein structure, depending on what protein you're talking about. So the primary structure is the amino acid chain. So this is simply the sequence of amino acids. The secondary structure is, there are 2 possibilities. One is the alpha helix, or the other is the beta pleated sheet. And whether an amino acid sequence will form either an alpha helix or a pleated sheet depends on hydrogen bonding between the amino acids within the sequence. Depending on where what the side chains of the amino acids are and where they're located, regions of an amino acid sequence can form one type of secondary structure, while another region can form another type. And there are also potential for disordered regions as well. And then tertiary structure is a secondary structure that is formed by disulfide bridges. So you may remember from the first session, there's one amino acid called cysteine that has sulfur in its side chain. So these disulfide bridges are formed by 2 cysteine residues forming this bond. And then coordinating structure, that doesn't apply to all proteins. This applies to proteins that are comprised of multiple subunits. So this essentially just means multiple tertiary structures forming a complex together. So, so how a protein folds is can be dependent on, it's dependent both on the side chains as well as the environment that it's in. So for instance, if you have both polar and non-polar side chains, and you're in hydrophilic or aqueous environment, the hydrophobic regions, or the which are the non-polar side chains, will be folded into the interior of this protein. Meanwhile, the polar side chains will be coating the outside of this protein. So that's if it's in an aqueous environment such as the cytoplasm. You could have the reverse, where a protein is in non-polar environment, such as the lipid membrane. So in that case, you would have domains of a protein that have non-polar side chains, which are embedded inside the membrane, and then regions that are that contain polar side chains can be exposed to the cytoplasm or exterior. So this is how proteins with what are called transmembrane domains are folded. Okay, so I'll get into more detail about protein folding next week. We're going to be talking about misfolding and chaperone proteins as well. But until then, are there any questions about protein structure?
I have two questions. With the schematic representation from secondary, tertiary, quaternary, is that just a schematic, or does it actually happen in that sort of stepwise way? Or does it all happen at once? I mean, when
the folding starts as soon as translation it has initiated, so it doesn't happen stepwise, but yeah, this is more just for schematic purposes.
To break it up into all the four structures that are within this one. The other question is this process passive, or does the cell have to spend energy to make the folding happen?
So it depends on the protein and the environment. So there are certain cases where what's called a chaperone protein is necessary to allow for a proper protein folding. This can happen when you have a very hydrophobic protein that's being formed in the cytosol. So then the chaperone basically gives it an environment where it can fold properly, and this is a process that requires energy and ATP hydrolysis. But it can also happen passively as well, because it's really dependent on the side chains and the environment that's in.
Can you define side chains again? How are side chains different from the order of amino acids?
So the side chains are, so every amino acid has an amino group, a carboxyl group, and then in between the two, they have, or branching off from it, it has what's called a R group or a side chain. So this is the one region of an amino acid that actually varies from amino acid to amino acid. And this is where its characteristics such as hydrophobicity or polarity, that's where it comes from.
That's where all the action is, in the side chain.
Is it right to think about, is this the functional parts of the protein, are those always on the protein surface? Or is there anything going on in the middle?
It depends on the protein. A lot of times the active site, especially with enzymes, they have what's called an active site. A lot of times that's in the interior, so that it's for regulatory purposes, that's generally how it makes the most sense.
I guess it depends on how you define inside and outside, right? Because they have very complex structures, like the RNA polymerase is like a claw, like a crab claw. And
Mhm.
so,
So what's inside? What's out? Yeah.
yeah, the DNA feeds into the crab claw, and this is where the copying takes place. Yeah, and other yeah. So it depends.
Is there such a thing as like a, I guess it would be called like pentenary structure or something, where proteins start to form complexes with other proteins?
I don't know if it's called that, but
But it does happen.
They just
yeah, I
call them
think that at some point you
complexes.
just start calling them complexes.
That's right. Yeah.
Or so those very complicated mixtures of RNA and protein. The ribosome is one, and then there's the spliceosome, which does the splicing of the mRNA. It's like lots of components. Oh, RNPs, ribonucleoproteins, right? It's amazing stuff if you think about it. It's just so intricate and endlessly complex. Just keep on adding on more and more bits and pieces. Yeah, magic. It's like magic.
Yeah, let's take a pause on the recording and maybe take a little break, or we can continue.
Okay, so now we're going to get into some protein-related techniques. So, first of all, I want to talk about what techniques we're going to go through. So, I'm going to talk a little bit about protein purification, which is getting a specific protein of interest out of the cell or out of tissue or an organism. Then I'm going to talk about ways to visualize proteins, starting with separating proteins on type of gel that's similar to what I talked about a couple of weeks ago about DNA gels. And then I'm going to talk about a couple of types of immunodetection, which is the use of antibodies to visualize specific proteins. So one way of purifying a protein of interest is through chromatography. So this uses so the example I'm showing here is called column chromatography, but there are other forms of other ways of performing this. So this uses beads that are made of either separose or agarose. They're a type of gel, and they're usually conjugated to either an antibody that's specific for your protein of interest, or some other feature of the protein is going to be, it's going to give it some kind of affinity to these beads. So an example that I sometimes work with when in my class is this image is actually from purification of green fluorescent proteins, which is it's a very hydrophobic protein. So essentially the beads here would give a hydrophobic surface for the protein of interest to bind onto. So that's one type of chromatography. Another type is immunoprecipitation, where the beads are conjugated with an antibody that detects the protein of interest and binds it. But the ultimate goal here is to have a specific protein bound to these beads. So how this works is that you have the cell lysate. So you can break apart your cells either using detergents or by mechanically breaking them open, depending on what type of cell it is. If it's a plant cell or something with a cell wall, usually you have to also do some kind of mechanical breakage. If there's no cell wall, sometimes a detergent of some kind is enough to break the cells open. Then the cell lysate is passed through this column, and this is basically a fluid that passes through the column. And ideally, your protein of interest should bind to the beads. All other components of the cell lysate should pass through. And then this column is treated, the column or the beads are treated with a series of wash buffers. And these are just buffers with specific salinity that will cause anything that isn't tightly bound to the beads to pass through. And the goal here is just to purify your protein further. And the last buffer that goes through here is called an elution buffer. So this varies depending on what type of affinity chromatography is being performed. So in the case of hydrophobic interaction chromatography, it would be a low salt buffer. So the amount of salt in the buffer will affect whether or not the protein of interest is able to bind to the beads or not. Other types of chromatography use some kind of a competing agent that will bind to the beads and displace your protein of interest. But the goal here is to get your protein off of the beads and into this eluate. So there are many reasons why we would want to isolate proteins. A lot of it involves scientific experiments, such as testing enzyme activities in vitro settings. It can also be performed to produce large quantities of an enzyme, such as Taq polymerase, which we talked about in PCR. Also be used for medical treatments. For instance, recombinant yeast can be used to produce insulin, which can then be purified from the yeast cells. Similarly, lactase digestion enzymes can also be produced in the cells to then they can be used by lactose intolerant patients. In addition, it can also be used in food production. So one example is that the enzyme rennet can be purified in order to make cheese. So this slide is from an old colleague, and she was just obsessed with making cheese. We actually had a session in the lab class that we taught where we would actually have the students make cheese. It was an interesting class. Specifically, we were making mozzarella. It actually turned out pretty well. Okay, so the next topic is, well, any questions about protein purification, first of all? Okay, I'm not going to talk about how to separate proteins by size by gel electrophoresis. So I talked a little bit about gel electrophoresis in the context of DNA. So when we were talking about that before, the idea was to use agarose to make this matrix, and then DNA would run through it. And because of the porous nature of agarose, the DNA fragments would be separated by size, because smaller fragments of DNA would be able to pass through the matrix faster than larger fragments of DNA. So it's a similar concept with proteins. So in order to prepare the proteins for gel electrophoresis, they first have to be broken down to their primary structure. So this is done by addition of a reducing agent called DDT and heat, and this breaks apart the disulfide bonds that are formed by those cysteine residues. And then the heat also breaks down the hydrogen bonds. So you're left with this primary structure, which is just the chain of amino acids. And then in this process, detergent called sodium diacetyl sulfate, which I'm going to refer to as SDS from now on, is added. And this will coat the amino acid chain with negatively charged ions. And the reason for this is that in gel electrophoresis, you have a positively charged end and a negatively charged end, and you're basically going to separate the molecules using electrical currents so that they'll all head in one direction. So this is a schematic of what a protein gel setup looks like. So SDS page stands for sodium didecyl sulfate polyacrylamide gel electrophoresis. So this is similar to agarose gel electrophoresis, which is what you do with DNA, but here the gel is made of acrylamide. This gives us a smaller matrix, of course, and the reason for this is the reason why we want this is because amino acid chains are smaller than DNA strands. On the top here, you have a negative electrode passing through the apparatus, and you have wells where the protein sample is placed. Then when it's turned on, the positive electrode is at the bottom here, and the proteins which are coated with a negative charge at this point are, they travel from the top to the bottom. And similar to what happens with DNA gels, the smaller proteins will travel faster because they're able to pass through the matrix more easily. What you have at the end is proteins that are separated by size. And then the proteins can then be visualized either by gel staining or by further processing by Western blot, which is a type of immunodetection. So the example of gel staining is Kamasi blue, which I'm showing here. So what this type of staining does is it visualizes all the proteins in the sample. So here, you may be able to visualize your protein of interest, but it can't definitively be identified. Here, really the only indication of whether whether you're looking at the protein you want to focus on or not is this molecular weight marker that's on the far left here. So this is similar to DNA ladder. Basically, these lines represent different sizes in terms of kilodaltons. And ideally, you would go in knowing what size your protein, the protein you want to focus on is, and you would be able to have a sense of where on the gel your protein is, but you wouldn't be able to definitively say that this is your protein. So in this case, this is one of my gels. I was looking for a glycogen synthase kinase, and this band here corresponded to the size, but without doing immunodetection, you can't conclusively say that's what the protein is. So one technique for immunodetection is called the Western blot. So Western blot uses a type of membrane, usually it's nitrocellulose, and basically what you do is you transfer the proteins from this acrylamide gel and you get it onto the membrane. And the reason for this is that the gel is not a stable surface for the proteins to be on. The nitrocellulose membrane is much more stable. This transfer occurs in an apparatus which contains a series of filters and padding to basically just give the membrane and gel some stability. And then again, it's set up with in a apparatus that has an anode and a cathode. And the proteins are will again move from the gel to the membrane in the direction of the anode. Again, this is because the proteins are coated with negative ions because of the SDS step in denaturation. This is just a summary. So all the proteins from this gel are transferred onto the nitrocellulose membrane, and then an immunodetection assay is performed to visualize the protein of interest. This involves the use of antibodies. Just a quick review of how antibodies work. So if you have an antigen, it provokes an acquired immune response. This can be triggered by bacteria, viruses, toxins, allergens, fungi, foreign DNA, or foreign particles. The antibody is a type of immunoglobulin protein that's formed in response to some kind of an immune challenge. So there are many types of antibodies, and they bind to specific antigens, and these antigens are then flagged for destruction. One strategy that a lot of scientists use is that they modify the sequence of a protein of interest to include a well-studied antigen sequence onto there at the end of the protein. So for example, the one I showed you before, here, this indicates that this protein glycogen synthase kinase was tagged with a protein A antigen, which means that the DNA sequence for protein A was added to the end of the glycogen synthase kinase sequence on the DNA level. And so when this is translated into a protein, the protein up until the end is just normal glycogen synthase kinase, and then at the end it has a stretch of amino acids that corresponds to an antigen known as protein A. And the reason for this is there's a lot of commercial antibodies specific for protein A. So rather than having to purchase antibody specific for glycogen synthase kinase, which would be really expensive and a labor-intensive process, you can just purchase antibody for protein A, and it would do the same job as far as detecting the protein of interest is concerned. So the process by which these antibodies used in immunodetection assays are generated is there are host cells which are stimulated to produce antibodies. These are usually done in small animals such as mice and rabbits. These host cells are then hybridized to tumor cells. These hybrid cells then proliferate quickly in a culture to produce a large number of antibodies, and the antibodies are then harvested and isolated for research. So an immunodetection assay uses these antibodies on the nitrocellulose membrane. So you start off with a nitrocellulose membrane that has all your proteins on it. So your protein of interest should be on the membrane, and at this point it's not visible. So to the membrane, you then add what's called a primary antibody. This recognizes the protein of interest or an antigen that's attached to the protein of interest. This primary antibody is also not visible. Then the next step is a secondary antibody is added. This secondary antibody recognizes the primary antibody. It does not recognize the protein of interest. This is also not visible, but it's usually conjugated to some kind of enzyme or some kind of a fluorophore. And this enzyme is what's going to allow the protein of interest to be visualized. At the last step, some kind of an enzyme substrate is added to the membrane if the secondary antibody is enzyme conjugated. So this can be either colorimetric or chemiluminescent, it depends on what type of imaging system is used. But this substrate will undergo enzymatic reaction with a secondary antibody, and this will give some kind of a signal, either something colorimetric or something chemiluminescent. What's becoming more common now is secondary antibodies that are conjugated to a fluorophore, specifically long wavelength fluorescence or a near infrared fluorescence. This is pretty recent, but the reason for why this is preferred over this type of enzyme conjugated secondary antibodies is that visualizing a protein through this technique is limited by the amount of substrate that you use, and it's also limited by the enzyme itself. So if you don't have enough substrate and all the enzyme is used up, you may not get an accurate signal of how much of your protein is present. If this is attached to some kind of a fluorescent molecule, instead you'll be able to you'll have a more direct you'll be able to more directly gauge how much protein is present. But again, this is pretty recent, so most labs that I'm aware of still use enzyme conjugated antibodies. So this is a Western blot. Yeah.
Can I just ask you, if you're going to do the immunodetection assay, why do you have to do the gel electrophoresis step? Why couldn't you just do the immunodetection on the stuff that comes out of the columns?
So you can in a different type of assay, but the benefit here is that since they're separated by size, you'll also be able to identify the protein based on size as well. So you'll have some separation because sometimes an antibody will it'll react to more than one protein. This is especially common in like in-house made antibodies. Also, sometimes you have multiple proteins that have multiple tags and there's cross reactions there. If they're separated by size, then you have a better idea of whether you're looking at the correct protein or not. Okay, so yeah, so the image on the right here is a Western blot. It's identical to the image, the samples are identical to the one on the left. So again, this is the one on the left is a Kamasi stain. So that's all the proteins present on the gel are visualized. And the one on the right is a Western blot this was a membrane that was treated with an anti-protein A antibody and then treated with a chemiluminescent substrate. So here you only see the proteins that are tagged with the protein A antigen. So you can see an obvious difference here. In these first few lanes, you see a ton of protein signal by Kamasi blue, but you only see the one band when you look at it by Western. In addition, these light lanes at the end, you can see that there's a lot more of the protein A tag protein. So this gives you an idea of whether the protein's present or not, and it also gives you an idea of how much of the protein is present. So yeah, and now I'm going to go into another type of immunodetection, but before that, are there any questions about Western blotting?
From that last step, from post electrophoresis, post immunodetection, is it possible to somehow sequence the polypeptide chain, or have you lost that information?
It is possible. So it can be sequenced by mass spec, and usually the stage where you would do that is actually before you transfer it onto a membrane. I think it should. I'm not sure if it's doable after it's on the membrane, but I actually do this pretty frequently. So when I send a protein out for sequencing, what I do is I run it on acrylamide gel by electrophoresis. Then I stain it with Kamasi blue. Usually when I do this, I use like ultra pure proteins, so this type of confusion doesn't happen. Then I just cut out the bands of acrylamide, and then I just ship that off to the mass spec facility. Okay, so the last type of immunodetection assay I'm going to talk about is ELISA, or enzyme-linked immunosorbent assay. This is typically done in this type of apparatus. It's called a 96 well plate, which is then inserted into a plate reader to quantify signals. So the most basic. Sorry, did someone say something? Okay, so this is another type of assay that relies on antibodies to determine the presence or absence of an antigen. The most basic form is called direct ELISA, and here an antigen is coded onto the multi-well plate, and then it's detected, an antibody is added that's conjugated to an enzyme. Then if this antibody binds to the antigen, then once a substrate is added, it should give some kind of a signal, usually a colorimetric one. Another form of ELISA is called indirect ELISA, and this is similar to what I just described with the Western blot, where you have a primary antibody that binds to the antigen, and a secondary antibody that's conjugated to an enzyme that detects the primary antibody, but not the antigen directly. That a substrate is added to yield enzymatic reaction with the secondary antibody. Another form of ELISA is called the sandwich ELISA method. So here, rather than an antigen coating the plate, an antibody coats the plate, and that antigen is added to the well. The antibody that's coating the plate is referred to as a capture antibody. Once the sample is added, it binds to the antigen that it's specific for. Then a second antibody is added, and this is called a detection antibody. So this also binds the antigen. And then a third antibody is added, which is similar to the indirect ELISA. This third antibody detects the detection antibody. It's also conjugated to an enzyme, and then a substrate is added to yield a colorimetric signal. And the last type of ELISA assay I'm going to talk about is called the competitive ELISA. So this is an inhibition-based assay. So here, a sample is an antigen is added to coat the multi-well plate, and then a capture antibody is added, and then following that, some kind of competing molecule is added, and the competing molecule contains it's linked to an enzyme. So the concept here is that the same amount of this competing molecule will be added to all the samples, but depending on the amount of antigen in each well, the signal coming from the enzymatic reaction is going to be different. So this would give you a sense of the level of an antigen that's present in the sample, depending on how well the competing molecule is able to bind to the antibody.
Can you explain the reason for the sandwich, Elisa?
Yeah, so the logic here is that rather than having the antigen coat coat the plate, it would be an antibody that coats the plate, and this would just give you more specificity because the antibody would only bind to the antigen, whereas there may be additional antigens present if you're just coating the plate with some kind of a cell lysate sample. So this would essentially ensure that only the antigen that you're concerned about here would be bound to the plate. Okay, and just wanted to talk about one particular application of ELISA. So this is actually the technique that's used for serological testing for COVID-19. So we talked a little bit before about the PCR test, where RNA is extracted from a sample and then it undergoes reverse transcription and real-time PCR to determine if someone has viral RNA present in the sample or not. And this is used for detecting viral RNA during early and advanced infection. The serological test involves using a blood sample, which is then the serum is then separated and used in an ELISA assay, and the presence of antibodies against the virus are then detected. So the enzyme that they're looking for here is a human immunoglobulin protein. This technique is used to detect advanced infection or recovery or previous infection. Yeah, so yeah, that's just one example of a real-life application for ELISA assays. Any questions? Okay, so that was all I was planning on covering today. So yeah, we can just take some time to just if you have any questions that you come up with, you can ask now, or and if we have time, we can also watch the video that Jeremy posted.